Good morning, everyone, and welcome to the NINDS Biomarker Q&A webinar. My name is Rakonda Metley, and I am the Operations Coordinator. We will start off with a presentation of slides followed by questions and answers. Please submit your questions in the Q&A section featured below, and they will be answered at the end of the presentation. This presentation will be recorded and placed on our YouTube page in the next couple of days. I will be sending that link along with the presentation slides to everyone that has registered. Now, let me introduce our webinar panel. Dr. Carol Taylor-Birds, who is the Program Director, Dr. Ram Aruchachan, Program Director of Hill Pain Biomarker Program, Dr. Tremajiman Salu, Health Program Specialist, and lastly, Biomarker Program Consultant and Program Founder, Marianne Palimonter. I will now turn it over to Dr. Taylor-Birds. Thank you.
Thank you, Rakonda, and thank you everyone for joining the webinar today. So I'm going to go ahead and get started. And just to quickly sort of go over the goals of the program and the objectives of the program, this is a program that's in our Division of Translational Research at NINDS, and it was developed in order to help facilitate the validation of biomarkers and biomarker signatures that are ready for use in clinical trials in order to accelerate the therapeutic development and advance patient care. And below is just a diagram kind of highlighting the process of biomarker discovery through validation as outlined in the FDA's guidance on establishing biomarkers as tools for therapeutic development. So we have funding opportunities for the discovery, analytical validation, and clinical validation stage. And I want to point out that while we do encourage applicants to apply for FDA qualification in the later stage funding opportunities, it's not actually a requirement, but it is highly encouraged. So as I said, we have funding opportunities for discovery, analytical validation, and clinical validation. These are the active programs. And I'm going to be going through each of these in more detail in a little bit. The first one, PAR 19315, is our discovery one. And then we have both U01 academic cooperative agreement projects for the analytical validation and clinical validation stage, as well as U44 small business versions for those same funding opportunities. And all of these are clinical trial optional. So I also want to bring up that we have two notices that were just published this year. One, ROM is the point of contact for, and this is a notice of special interest for NINDS mission pain research. And there's a specific section calling out the need for identification of pain biomarkers or biomarker signatures that predict or monitor response to therapeutics or therapeutic mechanism. And so you can apply to PAR 19315, the discovery PAR, and cite this notice if this is of interest to you. Our Division of Clinical Research also published a NOCI on the notice of special interest to encourage the submission of applications focused on the development and validation of biomarkers for functional neurological disorders. And all of our biomarker PARs are listed in this NOCI, so you can apply to any of those and cite this notice in the application. And the point of contact for that is Kodun Lungu. So I'm going to take a few minutes to talk about what biomarkers are. And I'm sure many of you are familiar with this, but several years ago, the FDA and NIH developed the BEST glossary, the BEST biomarker endpoints and other tools resource, to define what the different categories of biomarkers are and help clarify how they're used for different types of tools. So biomarkers can be categorized as pharmacodynamic response, diagnostic, prognostic, predictive, susceptibility, risk, monitoring, or safety. And the expectations and amount of evidence to demonstrate them as useful tools for each of those uses may differ slightly. So one of the things that is critical about this program and all of the funding opportunities in this program and make it distinct from some of the standard research project grants, R01s, and that NIH funds, is that these are all specifically designed to help establish the evidence that you would need in order to qualify something as a biomarker for use in for a specific context of use. And context of use is really critical in all of the FDA guidance. And it's a concise description that specifies the best biomarker category and how the biomarker will be used in therapeutic development or clinical practice. So it's not saying, you're not just identifying a biomarker that's associated with the disease mechanism and that's it. You want to go beyond that and say, this is the biomarker we're looking for. This is the particular need and use case that we see it being necessary for, and then we're going to test it for that use. So the examples here, just a few examples are, you might have a predictive or prognostic biomarker to help stratify or enrich patient populations. You might have a biomarker to demonstrate therapeutic target engagement, like a pharmacodynamic response biomarker, and help inform dosing decisions. You might have one that would indicate risk for developing a disease or condition, or to monitor a disease condition or change in response to an intervention. And of course, to inform treatment decisions. So the reason we spent a few minutes on this and really emphasize this in the program is that we really want to make sure that when you're applying to these funding opportunities, you want to be really thinking about how this biomarker is going to be used. And you want to make sure that your study design is set up to test your biomarker for the context of use that you're proposing. So I'm going to go through just a little bit more terminology. Some of the way we refer to this in the discovery funding opportunity is we talk about initial clinical validation. And what we mean here is that you're going to demonstrate that the biomarker has predictive value for the outcomes of interest for the clinical condition or biological process of interest. But you want to see that in the clinical data or clinical population. And then analytical validation is to demonstrate the accuracy, precision, reproducibility of the test, how you're detecting the biomarker, and how well that test measures what it's claiming to measure. And then in the later stage, definitive clinical validation, we're talking about something that's comprehensively evaluating the normative ranges. You're going to establish reference values and distributions across the spectrum of individuals that the biomarker is expected to be used with for the context of use that you're defining. And then you want to make sure that the statistical measurements and thresholds for using the biomarker signature in the clinical trial or clinical practice decision-making, it really needs to be established as part of the definition of clinical, or as part of definitive clinical validation. So I'm going to talk now about the R61, R33 discovery PAR, PAR 19315. So this is a phased mechanism. The phase 1 can be between 1 and 3 years, and the phase 2 can be between 1 and 3 years as well, but the total project length cannot be more than 5 years. So applications can request funding above $500,000 in direct cost in any 1 year, but if you need to do that, you need to get prior approval. So you need to reach out to me at least 6 weeks before submitting the application, because we need to take that to leadership for approval to come in. So the phased mechanism means that there are milestones that you need to propose in the application that are go, no-go milestones where you're going to define quantitative metrics that need to be met at the end of the R61 in order to justify continuation into the R33 phase. And I will talk about that a little bit more in a minute. This transition occurs through administrative review, programmatic review, and we do work with you. You need to propose these in the application, and we do also work with you if you're going to be awarded to refine them before an award is made. And I also want to point out and just make sure that you're aware that the application for these, you do need to submit the application for both the R61 and R33 phase. So you've got a 12-page application, and the study design for both of those need to be included. So the entry criteria for this PAR, of course, you want to know, is there strong biological rationale supporting the concept of the candidate biomarker or biomarker signature? And for the context of use or uses that are being proposed, and are you going to fill an unmet need in neurotherapeutic development or clinical practice, right? So biomarkers, again, are a tool, so you want to define why this tool is necessary and how it's going to be used. And for the discovery PAR, you don't necessarily have to have one context of use already defined. You might propose a couple that you're going to try to then decide if the biomarker is useful for one or both of those by the end of the R61 phase. And of course, these are fairly complex projects. These are large teams with diverse expertise. And so we want to make sure that you're including research teams that have the knowledge and technical proficiency for the studies proposed. And that might include things like clinical expertise, disease biology expertise, statistics, the technical modality expertise for the method of detection, and so on. So the research objectives for the discovery PAR are to identify a biomarker or biomarker signature, and you want to evaluate the candidate biomarker or biomarker signature for the use proposed. And then you're going to develop a working hypothesis regarding the context of use. So again, you're going to want to test whether or not the biomarker works for that hypothesized context of use. And then you're going to develop and do the initial internal validation of the biomarker detection method, if necessary. So you can come in with a fairly exploratory project, excuse me, where you might be hypothesizing about a couple of different types of biomarkers that you're going to identify, or you might come in with something where you've already got a candidate biomarker, and now you're going to develop it a little bit more. You're going to develop the method of detection a little bit more, excuse me again, and go on to do the initial clinical validation test in the R33 phase. So going back to the go, no-go milestones, these need to be met for the transition. And some of the metrics that you might think about including as milestones would be things like the minimal acceptable performance characteristics of the detection method. So things like precision, accuracy, analytical sensitivity and specificity, dynamic range for the detection method, or model stability, those types of things. And then you also want to talk about the minimal acceptable association of the biomarker with the outcomes of interest. And you want to be pretty clear on what you're measuring the biomarker against. How are you, what outcomes are they? Why are they appropriate? And what's the degree of association for the relevant clinical endpoints? Things like the minimal acceptable accuracy, sensitivity, specificity for the primary outcomes, right? So those, again, should all be quantitative. And you're going to say, what's the minimal amount you need in order for this to demonstrate potential utility for that context of use? So switching over to the U01 and U44 cooperative agreements, for both the analytical and clinical validation, these are cooperative agreements. So these are also all up to 5 years. Again, budgets aren't limited, but you need to get prior approval if you come in for more than 500,000 direct costs in any 1 year. And you want to make sure when you're looking at the small business budget, there's information on the link below for the phase 1 and phase 2. So these can be up to 700 per year for 2 years. And up to a total of 3 million for 3 years. But you can request up to 1.5 per year. So for the cooperative agreements, these, instead of just milestones between the R61 and R33 phases, you need to propose milestones for each year. And again, these need to be very clear and quantitative. And these get evaluated programmatically each year as part of your progress report response. So for the analytical validation, the entry criteria here, again, you want to have a defined biomarker and context of use. This one, you need to have a specific context of use already identified. And of course, all of these applications need to be within the mission of NINDS. Has the biomarker or signature already undergone initial clinical validation for the context of use proposed? So you want to know and demonstrate that you have a biomarker that already has some clinical utility and that you can clearly describe that in the application. And you don't want to be developing a new detection method in the analytical validation. This should be something that's already established, and now you're going to do additional optimization to really refine and perfect the detection method. So the research objectives on those lines are to conduct the analytical validation to establish the technical performance of the method of detection. You're going to optimize and standardize the detection method across multiple sites. This is not, there are some cases you can have a scientific justification if there's a reason this isn't possible. So for example, in some rare diseases where that might not be feasible, but the expectation is that these should all be multi-site studies. And you should be rigorously evaluating the analytical and pre-analytical variables because reproducibility and reliability of the detection method is absolutely critical for the success of the biomarker as a research tool. And of course, by the end, you want to have established the specified technical protocol or a pipeline for the context of use proposed. So this would be ready to go into clinical validation and be used in the clinical setting. So in terms of milestones, you might include annual milestones for both performance and progress milestones. So these again should be quantitative, but you want to define, the target sensitivity, specificity, accuracy, precision thresholds that you need. What are the acceptable dynamic range that's necessary for the context of use proposed? You want to demonstrate the standardization across sites and define the acceptable amount of variation within and across sites or operators. You want to demonstrate data quality assurance and quality control standards. And you also can include progress milestones, things like enrollment of samples and data collection, establishing contracts, standard operating procedures, and consultation with the FDA if necessary. Now, finally, with the clinical validation PAR, these are 21058 and 059. Here again, is it within the mission of NINDS? The context of use has to be clearly defined. Has the biomarker or signature already undergone initial clinical validation for the context of use proposed? And of course, we also want to see evidence that the method of detection has also already been optimized and has undergone analytical validation. So the research objectives, as I sort of mentioned before, conduct the definitive clinical validation to establish that the biomarker or signature acceptably identifies, measures, or predicts the appropriate concept of interest for the context of use proposed, and determine the statistical threshold for how the biomarker will be used for decision-making within a large and representative patient population. So here, we expect fairly large study sizes, fairly diverse patient populations that should be more heterogeneous than you would see in the discovery phase. So milestones, these are somewhat similar, but you can also look at clinical sensitivity, specificity, accuracy. You establish how the appropriate cutoffs or thresholds for decision-making would be for the context of use proposed. Determine the positive predictive value or negative predictive value in some cases. Demonstrate standardization across sites data quality and assurance quality control standards. And again, the set up in such a way that they're quantitative. So it's not ambiguous about whether you've met them or not. And then again, with progress milestones, establishing contracts, SOPs, enrollment and sample and data collection goals. And you might want to consult with the FDA and submit a biomarker qualification letter of intent and see if you can get support from the FDA. So for all of these PARs, studies that are not responsive include development of therapeutic agents or devices as the primary goal, and then evaluation of clinical efficacy, effectiveness, or clinical management as the primary intent are not appropriate. So natural history studies that don't include biomarker identification and validation would not be appropriate, although you might leverage resources from ongoing natural history studies. Likewise, you might leverage resources from the clinical trials. And then you are required to include milestones and studies outside of the mission, of course, and the NINDS. All of these are grounds for withdrawing the application before review. And I'm going to add one comment here. So I think one of the things with the discovery PAR that we want you to sort of think about is that within the discovery biomarker space, you can be doing work where you're trying to understand the mechanism of the disease, biology or pathology, and you might be identifying biomarkers as part of that research. But if you're not interested in trying to get them into actual clinical use, and if you're more interested in understanding the disease, then you're probably better off going to an R01 or R21 study section. So general advice I like to give to everybody, you want to be really clear about what the unmet need is, because the goal here is to have tools that are going to be useful for clinical trials or clinical practice. And so you need to justify that the tool is going to be better than what's already out there. And part of being able to do that, it's good to really be upfront about what's the current standard and how is this going to be an improvement over what that is. For study designs, you want to think about, should the test or biomarker signatures utility for decision-making within the context of use proposed? The statistical analysis plan needs to be really clearly described and appropriate for the context of use. I think one of the things that we often see in the discovery PAR is that you might have applications that are applying for, or are describing a biomarker that might be used for many different things, but their statistical analysis plan or their study design isn't testing it for those. And so that's not the goal here. And you should consider the plan to identify and address sources of variability, because again, we're really thinking about reproducibility here. Since these are tools, you want to know that they're reliable. And so you want to be thinking about what are the sources of potential variability that need to be dealt with. And then finally, just to mention the study section. So all of these PARs are reviewed at NINDS and the NSDA and NSTB study sections. And one thing to keep in mind is that you're going to have a lot of different reviewers with different expertise. So these applications will generally have between 4 and 7 reviewers. And you're going to have people that have expertise in the modalities of the biomarker that you're looking at, the clinical disease area or condition, clinical expertise, biomarker regulatory or pharmaceutical backgrounds. And so you want to think about the fact that when you're describing the need and the approach, you want to describe that a little bit more broadly and generally than you would for an audience when you expect all the reviewers to have the same background. So, giving a little bit more background about the goals and the need in this area, but you still want to make sure that you're including enough technical detail for the reviewers who are specifically looking at your proposal for the technical feasibility as well. So that's my last slide. We have a lot of resources on the biomarker webpage here. There's some FAQs on here. There are some links to other funding opportunities. And there's some information on resources available at NINDS and links to information at the FDA that are all useful for applying to these programs. So I'd encourage you to look at those and then reach out to me or any of us if you have questions. We're always happy to talk with investigators about draft-specific aims, so we do encourage you to reach out if you're interested in applying. And I'm going to stop there and we will switch over to questions. Thank you.
All right, thanks, Carol. Just a quick reminder for everyone, please submit your questions in the Q&A feature below and not the chat feature. Okay, our first question, is there a definition of initial clinical validation?
Yes, so I actually had that up on one of the earlier slides. And so that's available, they'll be available in the slides. But here, demonstrating that the biomarker has predictive value for the outcomes of interest for the clinical condition or biological process of interest.
Okay, the next question, someone asked if the slides will be available soon. Yes, I will send the slides hopefully by the end of this week, along with the presentation. Next question, what is the difference between this and a phase IIB SBIR proposal?
This in the phase 2B, I am not involved directly with the SBIRs other than the ones that we manage here, the U44s. I don't know, Ram, do you have any thoughts on that one? Yeah, I'd say just email us or
Carol, sorry, I was trying to unmute myself. The question is about what is the difference between this and phase 2 SBIR. So phase 2 SBIR is that if you have a phase 1 and then you met the criteria, you will go to phase 2B, or if you have a phase 1 and then complete it, and then you can come back as a new phase 2B. So that's the difference between the U mechanism and the SBIR. So other than that, the milestones and everything is same as here, what Carol was describing.
Thanks, Ram.
All right. Can you please explain how biomarkers can be used for neurotherapeutic therapy development? Does this mean the biomarker is used in developing new therapies rather than for a clinical decision-making?
Yep, that's exactly right. So, for example, a pharmacodynamic response biomarker, where you might be looking at a biomarker that's within the line of a therapy. So, you might be developing a drug. You want to determine if the drug is engaging with the target that you're looking at, and you want to know whether or not, what's the amount of relationship to the dose of the drug with the target engagement. So you want to have a biomarker that can tell you how the dose of the drug is responding, or how the biomarker is responding to the dose of the drug to help you in your phase 1 or phase 2 clinical trials. Or for something like a prognostic biomarker where you're looking at patient stratification, you might be trying to tailor your therapeutic to a study population that is more narrowly defined and more likely to respond to the therapeutic that you're developing. So those are just two examples. And these are, excuse me, these are a number of studies out there showing that biomarkers are really help advance the rate of therapeutic development. I'm going to just mute myself for a moment.
Okay, the next question, it looks like it's a set of 3, so I'll read them one by one. Is it reasonable in the R61, R33 to pursue an unbiased proteomic screen to identify the biomarker signature, for example, somologic proteomic screen or serum?
Yes, yeah, you can definitely do that. So with the unbiased screening, the one thing with these, we want to encourage you to be thinking about what types of markers you're looking for. So even if you're going in with an unbiased proteomics approach, you want to be talking about what kinds of pathways you're expecting to see that are relevant for the disease. So why, what types of markers do you expect? It doesn't want to be a complete fishing expedition.
Okay. How can the R61, R33 transition be written when the signature elements are known up front? For example, whether there are clinical measures already FDA approved or not is not known. Can a non-FDA measure be carried into R33?
Yeah, so this gets at to why you would want to think about what types of markers you're looking for, because it does make writing the R33 phase a little bit more challenging. But you can, this is where context of use again is really important, because you want to say, here's how we're expecting the biomarker to be used. And even if we don't know what the marker is, these are the types of performance criteria. This is the level of association with, the clinical outcome that you're looking at, or change in the disease state that you would need to be able to show in the R33 phase. Now you can say, you might revise your power calculations and your analytical plan at the end of the R33 phase, but you still need to be outlining what your expected study design would be.
Okay, next question. For PAR 19-315, there's only one deadline remind. If I submit for February 22, 2022 deadline, will I have a chance for A1?
Yes, so this was approved at September Council for reissue. And when we send the slides out, we'll also send a link to NINDS's list of concepts that were approved at Council in September.
The line between analytical validation and clinical validation is a little vague. Both of them focus on defining PPV and NPV accuracy. Can you please elaborate a little more?
Right, so you can think about the analytical validation really needs to focus on the detection method, right? So you want to be thinking about your ability to measure the biomarker for the context of use and how it's being measured. With the clinical validation, you're now taking it and applying it as a tool for the use proposed. And you want to be thinking about it in terms of how it actually predicts, the predictive value for response to treatment, or how well it differentiates appropriate populations of patients for patient stratification. So the emphasis is on the clinical use versus the detection method. And we talk about these in generalities. Sorry, I'll just add to that. We talk about these in generalities because, biomarkers can have a really diverse range of uses and modalities. And so this is why it can be good to sort of reach out to us and with a draft aims page if you're not sure which one you're ready for.
Okay.
Carol, could I add something to that also?
Yeah, of course.
I think in terms of the parameters that you'd be looking at for clinical versus analytical, they are some are the same and some are different. For instance, negative and positive predictive value would not be looked for in analytical validation, since as Carol mentioned, that's more about the detection method. Instead, you would be looking for things like precision. You'd be looking for things like analytical sensitivity, i.e. how much sample do you need? How sensitive is the marker in terms of picking up very small amounts. You'd be looking for accuracy, whereas in clinical, you would be looking for things like sensitivity in terms of true positives, true negatives for specificity, receiver operator characteristic, area under the curves, negative predictive value and positive predictive value. So those are some of the differences even in parameters that you would see.
All
Thanks,
right.
Marianne.
Next question. NINDS NIH StrokeNet has RFA biomarker study. Is there any coordination between two groups?
Sorry, can you read that one again?
Sure. NINDS NIH StrokeNet has RFA biomarker study. Is there any?
I see.
Okay. Is there any coordination between the two groups?
Sorry. So we work across divisions quite a bit. They're reviewed in different study sections and the point of concepts are different, but we work with the Division of Clinical Research and the Division of Neuroscience and vice versa. And so there's a lot of discussion across funding opportunities.
It seems that R61, R33 for discovery phase U mechanism is for both analytical, clinical validation phases. Are these grants all reviewed by the same study section?
Yes, they are. So all of these are in NSDA and NSDB.
All right. Please clarify how much of the application 12 pages should be focused on the R61 and then the R33. My understanding is that the milestones of the R61 should be completed before proceeding with the R33. Therefore, most of the application should be about the R61. Is that correct?
You really want to outline both. So there's not a certain number of pages for one or the other. You need to decide based on what your studies are, how much detail you need for each. I think that's one where you have specific questions about draft aims page. Maybe we can discuss that offline because there's no hard and fast rules about that. It really depends on the study.
Okay. How will reviewers evaluate differently proposals addressing biomarkers for clinical use and biomarkers for the neurotherapeutic development? Should this be described in the context of use?
Yes, this would be part of your context of use. So you would specify if you're trying to use something, develop something for clinical practice versus therapeutic development. And if you're proposing both, then what would the different metrics need to be for showing it was useful for one versus the other?
Do you know whether PAR 19.3.15 will be extended beyond February 2022 submission cycle?
Yep, I think we answered that one already.
Oh, okay. Okay, next. Do these mechanisms give special consideration for early stage, early career investigators?
No, unfortunately, that is not part of the expectations for these funding opportunities. So with the R01s, many of you know, if you're an early stage investigator, then there's an extended pay line that gets set for those. But because these are not pay line driven PARs, you don't have a particular advantage being an ESI. I will say for the R61, R33 phase projects, one thing you want to think about if you're an early stage investigator is that if you successfully complete the R61 phase and transition to the R33 phase, those are then considered R01 equivalents. And so you would no longer be an early stage investigator. But it wouldn't be until the R33 phase. So just something to keep in mind.
If you are pursuing a PD biomarker, do you have to be able to show an actual dynamic response during the R61, R33 grant, or is it sufficient to show that the signature is measuring the disease state?
Yeah, so the context of use there, you might do the show the response to the treatment in the R33 phase, where you're trying to actually test it for the context of use, but wouldn't necessarily need to be done in the R61 phase.
Okay. Does R61, R33 affect ESI status? Is there a multi-PI option?
Right. So multi-PI option, absolutely. And I guess I just answered the ESI question before.
Yeah. Okay, that's what we have so far.
All right. And I know the
one just came in, sorry. Do you have to include animal data in validating a biomarker?
No. So animal data is not actually, animal studies are not allowed in the analytical and clinical validation PARs. It is allowed in the R61 phase of the discovery PAR, but it's really not encouraged. We want to see these move into clinical data samples studies as quickly as possible. So they need to be strongly justified if there's a reason for including the animal studies.
Okay, another one just.
Yeah, so I said this before, but it's NSDA and NSDB at NINDS.
Okay.
Sorry, I read that ahead of time. So which study sections these are at?
Okay, thanks.
All right, are there other questions or other comments from anyone on the team? Yep.
Okay, sorry. Would biomarkers in a 3D organoid model be relevant instead of clinical samples?
You might test that in the R61 phase, but it would not be appropriate in the R33 or later stage ones.
Okay.
Yeah, if you're applying to one of these funding opportunities, you should reach out to me. You and Rakonda will help set up a meeting. And if you're specifically interested in pain or heal applications, you can reach out to Ram. All right, it looks like we might be out of questions.
Yep, I'm not seeing any on my end as well.
All right, well, thanks everyone for attending. Please do reach out if you have additional follow-up questions and want to set up a call. And as Rakonda said before, the recording and slides will be going out after in the next few days. What are the likely grant funding ranges? And is this a 3-year proposal? So I'm not sure which proposal you're discussing, but in the R61, R33 phase, you could have 1 to 3 years for the R61 or 1 to 3 years for the R33 phase, but you're going to propose a 5-year project total. For the analytical validation, these are often shorter, so you can have up to 5 years, but you may only need 3 or 4. Yeah, okay. Sorry, Rakonda, you want to go ahead?
Sure. Can you describe the competitiveness of these grants? What percentage are typically funded?
Yeah, so because we don't have a set pay line These aren't reported out the same way, but they're roughly equivalent to the NIH pay line. And in terms of budget, to answer the previous question as well, these really range drastically depending on the application. So you might have something fairly small in a 250 range in the R61, R33 phase, or we often see projects in the, close to the 500K and occasionally above that. And the analytical and clinical validation, especially the clinical validation, tend to be more expensive.
Another one came through. If we investigate PD biomarker, can we use therapeutics?
Yeah, so this gets to the question about what's in scope versus out of scope. So the way this often works well is if you're collaborating with an ongoing clinical trial and you're testing the biomarker for the pharmacodynamic response biomarker in conjunction with that ongoing clinical trial. You can conduct a clinical trial with therapeutic. It's just the focus needs to be on validating the biomarker rather than demonstrating efficacy of the therapeutic.
Do you need clinical preliminary data for R61, R33?
No, you don't. No.
Okay, we have another one, Carol. In the analytical portion of the application, can we include other centers? And do we need to include an agreement between centers, or a support letter is enough?
You can certainly include other centers. We encourage multi-site study designs for the analytical validation. So having centers or support letters would be necessary, but it's good to have the agreements already established, but it's not required. All right. Give it maybe one more minute, and if we don't have any other questions, we will wrap up.
One more came through. Are the discovery phase, analytical, and clinical validation one grant?
No, so the discovery project is an R61, R33 phase, and then the analytical and clinical validations are U01 or U44 mechanisms. All right, any last questions? Give it one more minute. Any last comments? All right, thanks everyone. Take care and Happy New Year.